Blood is kind of fluid tissue, which circulates in the circulatory system and play an important role in material transport. What are the components of blood? What are the physical and chemical properties? With these questions in mind, let's look at this section. First, to introduce the composition of blood. A certain amount of blood is mixed with an anticoagulant and a centrifuge at 3000 rpm for 30 minutes. Due to different blood-specific gravity, the blood is stratified. The upper yellowish fluid is plasma. The lower deep red is red blood cell, and the middle white opaque thin layer is white blood cell and platelets. So we know that blood is made up of plasma and blood cells. The basic component of plasma is a crystalloid solution. Since solute and water can easily pass through the capillary walls, the amount of electrolytes in the blood is basically the same as in the tissue fluid. Clinically, the concentration of various electrolytes in plasma can roughly reflect the concentration of these substances in tissue fluid. Another important component of plasma is plasma protein. Plasma protein is a general term for various proteins in plasma. Plasma proteins do not pass freely through the walls of blood vessel. So the main difference between plasma and tissue fluid is the low protein content of tissue fluid. Plasma protein can be divided into albumin, globulin, and fibrinogen by sorting out method. Globulin can be further divided into alpha globulin, beta globulin, and gamma globulin by electrophoresis. Various plasma proteins perform different functions. The percentage of blood cell in the volume blood is called a hematocrit. It is about 40% to 50% in healthy adult men, 37% to 48% in adult women, and 55% in newborn babies. Since the number of white blood cells and platelets in blood is very low, which only account for less than 1% in the total volume, the hematocrit can reflect the relative concentration of red blood cells in the blood. The hematocrit in an anemia patient decreases. In patients with extensive vomiting and diarrhea, plasma volume loss can result in relative elevated hematocrit. The physical and chemical properties of blood include 4 aspects: specific gravity, viscosity, pH, and osmotic pressure. Focusing on osmotic pressure. First, let's study the concept of osmotic pressure. Solute osmotic pressure is the force of solute in a solution to attract water molecules through the semi-permeable membrane. The characteristics of semi-permeable membrane is that it allows the solute molecules to pass through, but not solute molecules. Look at the picture. This is a U-shaped tube with a semi-permeable membrane in the center at the bottom, water on the left, glucose solution on the right. At the beginning, both sides of lipid level is flat, but after a period of time, the lipid level in the U-shaped tube changed. Water molecules flow through the semi-permeable membrane to glucose solution side, and the liquid level appear low on the left and high on the right, which is the seepage phenomenon. Water flows from the low permeability direction to the high permeable direction under the action osmotic pressure difference. The osmotic pressure depends on the number of the solute particles in the solution and is independent of the type of solute and the particle size. Plasma osmotic pressure is the osmotic pressure formed by solute molecules in plasma. The osmotic pressure is about 300 MOSM plasma. Plasma osmotic pressure is divided into crystal osmotic pressure and colloidal osmotic pressure. Due to the large proportion of crystal substances in plasma, plasma osmotic pressure mainly comes from crystal osmotic pressure, accounting for 99.6%. Crystal substances mainly consist of sodium ion and chloride ions. Colloidal osmotic pressure is mainly composed of albumin in plasma protein, accounting for only 0.4% of plasma osmotic pressure. Most of the crystalline materials in extracellular fluid do not pass through the cell membrane easily, and the crystal osmotic pressure inside and outside the cell remains relatively stable under normal conditions. Once the crystal osmotic pressure in the extracellular fluid increases, water molecules will leak from the cell to the outside to the cell, and the cell will shrink due to the lack of water. On the contrary, when the osmotic pressure of the extracellular fluid liquid crystal decreases, water molecules will enter the cell from outside to the cell, causing the cell to expand or even break. Therefore, plasma crystal osmotic pressure play an important role in maintaining the water balance inside and outside the cell and maintaining the normal morphology of the cell. Plasma protein is not easy to pass through capillary wall, so there is a colloidal osmotic pressure difference inside and outside the blood vessel. Therefore, although plasma colloidal osmotic pressure is low, it can retain water in the blood vessel, which play an important role in maintaining the water balance inside and outside the blood vessel and the normal blood volume. Clinical liver diseases is terminal, often accompany edema to happen. The reason in liver function drops can bring about content of plasma albumin to reduce, make plasma colloid osmotic pressure drops, broke the water balance inside and outside blood vessel, make water accumulation into tissue and appear abdomen. Additional kidney diseases or malnutrition can also drop because of the plasma colloid osmotic pressure and appear edema. Summarize, blood is fluid tissue made up of blood cells and plasma. Blood cells include red blood cells, white blood cells, and platelets. Plasma is a crystalloid solution. Plasma crystal osmotic pressure play an important role in maintaining the water balance inside and outside the cell and the cell morphology, while colloidal osmotic pressure plays an important role in maintaining the water balance inside and outside the blood vessel and blood volume. Thank you for listening. Goodbye.